So as has been mentioned many times throughout the symposium, understanding the neural circuit, the compound neural circuit, is one of the most essential neuroscience questions right now. And even the same pictures have been shown many times. We already knew more than 100 years ago that the neural circuit is very complex. The brain has the fortune of having a huge range of components or parts that it can use to build complex circuits to perform all kinds of complex and challenging tasks. So how to understand the function of a circuit? I would not say right now that the genetic approach is the ultimate approach to really understand the circuit. One main reason being that it still cannot be used for the human brain to try to understand the human circuit yet, at least at this time. So maybe some smarter ways will come up in the coming years, and we certainly hope that will happen. But just for the time being, at least, and for even the foreseeable future, there's no question that using genetic approaches in model organisms such as mice is probably the most viable approach to try to understand the components of a circuit and how each component contributes to the overall function of the circuit. So then, because of that, there's also no question that at this time, there's absolute need of having more and better genetic tools to be able to label and manipulate different kinds of cell types within the circuit. It's really a good time to do that now, given the huge advantage that has been achieving over the last few years. As Sasha Nelson was mentioning, when he started to do cell typing several years ago, there hasn't been so many tools that are available to do all these things. But at the same time, there's also this, with rapid technology advancement, we will be able to do a lot more. The second aspect is that exactly because of the rapid development of different genetic tools, it's also now the time to use the tools in a systematic way to understand the different cell types and the function of different cell types in a circuit. And as Christoph mentioned in his introduction, we are fortunate to also have Paul's support to possibly doing large-scale efforts in both two aspects. Today I hope to touch upon those two issues in some of the work that we have done recently. So here is just really a quick summary of the kinds of different genetic manipulations you can do to the cell types and the role of cell types in circuits. You can look at the morphology and the structure of individual neurons or neuron types by fluorescent labeling, just a general fluorescent labeling or subcellularly targeted fluorescent labeling. You can look at the connectivity between different cell types using a combined genetic transgenic mice approach with a viral-based tracing approach. You can look at the in vivo response properties of different kinds of neurons using genetically encoded sensors, such as calcium sensors or voltage sensors. And finally, you can also manipulate the function of different cell types using activating and inactivating genetic tools. So like many other groups, we have started a systematic effort to generate different kinds of Cre lines to target different cell types. And here is just an example of some Cre lines that we have generated. We started with taking a back transgenic approach, like the GenSet project. Recently, we have been focusing a lot more on a straight knock-in approach, not only for to be able to get more faithful recapitulation of the different cell types, but also we can build in some genetic strategies so that we can utilize the exact same loci for other purposes, which I will touch upon later. So, and I actually will start with talking about utilizing the different kinds of Cre lines to do a systematic large-scale work. And this is exemplified by this new large-scale project that we have started almost 2 years ago now, which is to use a genetic tracing approach to map mouse-brain connectivity. And the basic idea of this project is to, the main idea is to use different kinds of Cre lines that label different cell types, and then using a Cre-dependent viral tracer such as AAV to inject into specific regions, and that gets infected into the specific neurons that are marked by the Cre driver, and then map the axonal projections of those specific group of neurons throughout the entire brain. So the pipeline conceptually is quite simple. Stereotaxis injections of the viral tracer into a particular brain region. And then the viral tracer can express in fluorescent proteins at a very, very high level, which affords a high throughput fluorescent scanning process that we can capture the axonal projection information throughout the entire brain in a systematic way. And then all this fluorescent scanning data can be incorporated into a common 3D space, and projection maps, computational models, and things like that can be built upon that data set. So why doing a mouse-brain connectivity atlas at this time and on such a global scale? There has been a lot of knowledge about mouse-brain connectivity, general brain connectivity, but all the existing knowledge has been scattered around in the literature, and it's not complete for a given species or organism. The data are coming from multiple species, from primate, rat, or mouse, and the data are not captured in a standardized way. So what we are hoping to do is to use a standardized high-throughput pipeline to be able to acquire the whole brain projection data set in a systematic way, and also then display all the data in a public online website, just like the Allen mouse-brain gene expression atlases. And also we can link the different databases together. So present all the data in one place in a comprehensive way. This particular approach can, of course, is also scalable. You can, once the system is set up, once we finish the first round of high throughput whole brain level mapping, we can easily add more components to refined anatomical regions, or to use newly generated Cre lines that target even more different cell types, or have even more improved cell type specificity, or to study disease models, mutant mice and things like that, or to add transsynaptic labeling or other kinds of techniques to probe, connectivity in a more detailed fashion. And finally, this genetic tracing mapping approach is also reproducible. The first step not only is the anatomical tracing, but also it can, by switching the AAV viral tracer, you can rapidly move from structural study to functional study. And so you can combine circuit mapping with gene expression profiling to understand, of course, what kind of molecular or genetic signatures that the neurons have. That's connected to a specific projection pattern. You can combine this kind of large-scale whole brain level mapping with higher resolution synaptic detailed mapping. You can look at the monitor neuronal activities in a circuit with functional indicators. And finally, you can manipulate or probe the functional interactions using activity silencers or activators. And one good way that you can do this, we're thinking that You can devise, for example, you can devise very targeted slices that can capture an entire circuit from the originating site to the target site, and being able to preserve the integrity of the circuit and then probe the interactions of the two different connected regions in a well-preserved way. So for the basic components of the Connectivity Atlas project, we'll start with doing a non-Cre-dependent, just a regional specific mapping using regular AAVs as tracers. And we would target 300, 500 injection sites that covers the entire brain. And compare this kind of data with conventional tracer to validate the approach. After the first initial phase is done, we will be moving into the primary component, which is cell type specific projection mapping. And it will be combined with different kinds of Cre lines and Cre-dependent AAVs. Our current plan is to use at least 100 different Cre lines labeling different cell types throughout the entire brain. Of course, the more Cre lines, the better. The one key issue for this project is to be able to image so many different brains in a high throughput way, while at the same time capturing high resolution information. So the system we have adopted is, which Christoph mentioned yesterday, is able to capture fluorescent images at about 0.3 micron resolution in the XY dimension. And then to speed up the capturing process, we're actually going to do 100 micron sampling density overall throughout the entire brain. So there will be a lot of gaps in between sections, but we think that in this global scale, the data would be sufficient to capture the big picture. Of course, we always have the option of doing denser sampling around injection site and target areas by supplementing with confocal imaging or something like that. Given the current technology, we actually have been able to achieve the desired throughput, which is imaging one brain per microscope per day. And with this kind of scope and multiple units, what we are hoping to do is to be able to image thousands of brains, maybe more than 3,000 brains, all together in the next couple of years. So there will be multiple data modalities incorporated into this database. And first of all, we will characterize all the different Cre lines that we will be using in the atlas. And this is ongoing work, and we have actually done a substantial amount of work in this. The characterization will be mainly done using our regular in situ hybridization platform. After the Cre pattern is confirmed, they will be tested using the Cre-dependent tracers, make sure everything is correct. And then when that is done, the particular Cre mice versus virus combination will be put into this high throughput fluorescent imaging platform, and whole brain data will be collected. At the same time, as I mentioned earlier, we'll have a small-scale BDA versus the conventional tracer BDA versus AAV comparative study to validate the AAV as a tracing approach, and also to see the pros and cons of the two different approaches. Optionally, we can, as I mentioned earlier, we can do high-resolution imaging. Finally, we're also building a set of reference data sets so that we can use for better anatomical annotation of the axonal projection data. And all this data will be incorporated together into a common 3D reference space so that the starting site as well as all the projection site can be accurately mapped and 3D models being generated. So to do all this work, we have been carrying out a large number of pilot studies, as well as activities to establish the entire pipeline. And this is some of the activities are summarized here. We have been optimizing the AAV tracers to achieve really strong labeling so that the native fluorescence is sufficient to visualize all the axonal fibers without a need for doing laborious immunostaining. We have also optimized our injection method to achieve small, precise, and reliable targeting without much contamination. And we have also optimized our automated fluorescent imaging technique to achieve desired sensitivity, resolution, and speed that's needed. And we settled down on the different imaging platforms for both slide-based scanning as well as this whole brain Blockface imaging, which is one brain a day. And of course, the pipeline has been optimized, and data processing and web display of the data is ongoing. As I said, this project is really trying to take advantage of all the different kinds of genetic tools that are available out there, not only the ones that are generated from us, but also from the entire community. And we are able to obtain many different wonderful Cre lines from different resources. This include the GenSet project that Nat Hines talked about, the NIH Blueprint Cre driver project that I think Sasha Nelson talked about, as well as many of the individual investigators who have been generating different kinds of Cre lines for different cell types. And here I just wanted to show you some data, how things are progressing. Here is just some example of showing a comparison of BDA with AAV using a co-injection or serial injection method. It shows that the data now is, of course, it's not Cre-dependent yet. It's just regional specific. But it's here it shows that the both tracers are able to label the same site precisely. And if you look at the target areas, the AAV-labeled axon fibers versus BDA-labeled axon fibers, they do not completely overlap on a single fiber level, but they are largely confined in the same area. And this is true for both examples shown here. And here is an example of some pilot studies on actually Cre-dependent labeling using a particular Cre line, NR5A1 Cre, which has very specific Cre-induced gene expression in the hypothalamus, this region called ventral medial hypothalamic nucleus, and as well as the layer 4, cortical layer 4. So we've tried a combination of Cre-dependent virus with a Cre-independent virus and targeted those two regions. As you can see, the injection is very precise. However, with the help of the Cre line, the cell type specificity and the regional specificity is even further improved. And here's just a blow-up picture showing that is the case. The GFP green is the Cre-independent virus, and the Cre is the red virus is the Cre-dependent. So you can see that even though this injection is very precise, targeted to the region, the Cre line is still able to confine the infection and expression of the viruses in this particular region, whereas the non-Cre-dependent virus would spill over into the neighboring nucleus. In this case, in the cortex, non-Cre-dependent virus would label throughout the entire cortical column, different layers within that cortical area, whereas the Cre-dependent virus would be confined to the layer 4 neurons. And then here is several examples that I want to show you. And the 2-photon whole brain Blockface imaging system that we have recently set up and validated and beginning to generate lots of data. In this particular case, you can see that the scanning systems go through the entire brain at 100 micron sampling densities, generating 140 sections covering the whole brain. And as you can see, the virus is injected in this particular region, primary somatosensory cortex. And all the axonal projections can be captured in this entire series. And here I have one enlarged view to show one section that contains the injection site. This particular injection, it's actually targeted more to the deeper layers. So the layer 2, 3 neurons are not fully labeled. But nonetheless, you can see the axonal fibers going into contralateral cortical areas as well as subcortical areas. And I have a movie for this particular series. I hope to be able to show it at the end. This just has some enlarged views of that same brain. Of course, with fluorescent signals enhanced, exuberant axonal projections in contralateral cortex, in superior colliculus, and in different nuclei of the thalamus. And here's another example with viruses injected into the primary motor cortex area. And the projections coming out of that region throughout the entire brain is just mind-boggling. So there will be a series of sections going from the front of the brain to back. It's not an entire series, it's just representative sections. This is not even the injection site. It's a few 100 microns before the injection site. So you can see this extensive axonal and dendritic arborization near the injection site, but also a huge amount of projection into the contralateral cortical areas. And I think it will just gradually fly. This is the section that actually contains the injection site. This is the one later, just going the other side, going to the striatum. Extensive labeling into the thalamus, again, in thalamus. And then it goes all the way back to inferior colliculus, and all the axonal fiber tracks are going all the way back into brainstem, and then extensive branching in various brainstem nuclei. And here's another example, which is an opposite example with very specific targeted projection. This is a virus injected into dorsal LGN. So the projection is supposed to be specific to the visual cortex, and in this case, it looks like very much concentrated in the primary visual cortex. The section is again moving from the front to the back of the brain, and in this section, you don't even see the injection site yet. Seems like you see this is the fiber tracks that coming out of the DLGN. It looks like the fiber tracks going DLGN and then go out, go more anteriorly, and then turn around more anteriorly into this section, and then turn around through the striatum, and then back into the cortex again. So that's the injection site, very small and concentrated, and this is part of the cortex. And again, see that here. So this section is the center of the projection target zone. As you have heard, the thalamocortical projections into primary sensory areas, the V1 is heavily focused on layer 4 and layer 3, massive density of axonal terminals, with some projections terminating in the deep layers as well, especially layer 5 here, and a little bit, for this particular injection, a little bit in layer 1 as well. Okay. Final example. This is, so what about a visual cortex itself? Because we've been talking about this a lot. So I just wanted to show this one example. Again, this is a series of sections. I'm actually going to click through the massive projections that you see from this viral tracing. This is, again, a little bit posterior to the injection site. It's not exactly the injection site, but you can already see the huge amount of dendritic arbors and axonal arbors near this area, as well as spreading throughout this entire posterior visual cortical area. And the sections a little bit ahead of that. That's the injection site. But the massive local projections within that local area. And then you see it branches out into more ventral areas, maybe TEA or other associational areas, as well as go to the contralateral side and subcortical regions, superior colliculus. It just keeps going. And you see this axon fiber bundles going this way. They actually go this way and then go back up into inferior colliculus and superior colliculus. So here's the section that captures axons going into inferior colliculus and then up also to superior colliculus. And also you see the projection into other associational visual areas. This going further anterior to the brain. I see the axons into striatum and into this lateral posterior nucleus of the thalamus. Very dense, heavy projection here. And it keeps going with the different kinds of association area. I'm tracing those very well, those regions, where those regions are. And you can see the closal axon fibers going into other regions. Not only those major target areas can be captured very well, even all the minor ones, you can see them very well as well. So for example, here, the contralateral side, I think this is perirhinal cortex, is some sparsely labeled fibers. You can see them quite well. And then going even further anterior to the brain, this kind of domain, dorsal medial domain in striatum. And this tiny little bit projections in this area. Even further more into the frontal cortical area, some labeling in the anterior cingulate cortical area. And finally, even further front into the orbital frontal cortical area, you can still see lots of axon fibers. So this is just examples, several different examples demonstrating the intense labeling and specificity of the projectional mapping throughout the entire brain. So I just wanted to use the last few minutes to go from using the genetic tools to do a large-scale work, connectivity mapping work, into building some of the genetic tools. And as I said, we have an ongoing effort in developing different kinds of Cre lines for cell type specificity. I want to briefly mention we have also two other efforts in building different kinds of genetic tools. One is the Cre-dependent, of course, effectors that can express all different kinds of manipulating or visualizing tools that can be used in conjunction with a whole range of Cre mice for manipulation and monitoring. And finally, developing the next generation intersectional strategies so we can subdivide cell types or doing even more refined labeling of some complex cell types as many people have talked about, such as what Sasha Nelson talked about. Build on the very early success that we had in generating a whole set of fluorescent marker expressing Cre reporter lines, which Mike Stryker just mentioned, the AI9 and AI14-9. We heard from Jax that actually the AI9, AI40 TD tomato reporter line just over the last 2 years has become the top 3 most requested mouse line ever in their repository. So very powerful genetic tools. And here I'm just show you, we hope to surpass that. We'll someday become #1. So this is a new set of genetic tools is optogenetic tools that we use the same strategy, but we demonstrated for the first time that all these optogenetic tools can actually also work very well in transgenic mice in combination with different Cre lines labeling different cell types. So in this particular example, we had 2 different channel rhodopsin expressing reporter line, AI27 and AI32. This AI is our code, of course, for Allen Institute. AI27 expressed the channel rhodopsin link to TD tomato. AI32 expressed the channel rhodopsin link to EYFP. For whatever reason, the AI32 line is very, very super sensitive to light, and its sensitivity to light is comparable to in utero expressed or virally transduced channel rhodopsin expression. that's shown in this in vitro slice work. Also, we have data demonstrate that the in vivo activation of cortical neurons in awake behaving animals can be easily achieved using a normal light level that people use for in conjunction with channel dopsin expressing viruses. And here is another example showing in vivo activation of an inhibitory cell type, the hippocampal parvalbumin neurons, that in a very specific way. In this case, whether you use a regular light pulse or a sine wave light pulse, the parvalbumin neurons can be specifically and strongly activated, whereas at the same time, a nearby excitatory neuron has no response at all, and not only that, it's actually inhibited by the activation of the nearby parvalbumin neuron. And it's physiological studies showing the specificity of the light-activated neurons. That's exactly correlating the physiological properties of putative inhibitory neurons. So all this work is done, calculations done in collaborator labs, Karl Svoboda, Xue Han, and Yuri Busharki. And this is a set of inhibitory optogenetic Cre-dependent mice. This AI35 expresses Arch or Arch ER2 version of the silencer, and AI39 expresses the EMPHR 3.0 version of silencer. In vitro, both of them can very effectively mediate silencing of neuronal activation, and the light response curve is the Arch one is a little bit more sensitive than the EMPHR. And same situation in vivo in the behaving animals. The light can inhibit the cortical pyramidal neuron activity instantaneously upon the light on in both cases. And the Arch mice has the light sensitivity that's comparable, again, to a virally transduced Arch expression, whereas the EMPHR requires somewhat higher light level to be able to have efficient inhibition. And I have this one slide here showing our latest data on another reporter mice that we generated that expresses GCAMP3, the calcium genetically encoded calcium indicator. This mouse line is called AI38. And this is data, again, coming from the Janelia Gecki team. And many people know Hatim here. He used to work at the Allen Institute, but he did this part of work at Janelia together with Karl Svoboda and Cai-Wen Chen. Basically, I don't need to talk about the details of this work, just showing that the expression of GCAMP3 in cortical pyramidal neurons, layer 2-3 pyramidal neurons, is very good, and cells are completely healthy over 10 months of period of animal's life. The response, you can see many neurons with very good calcium responses. However, I should point out that the expression level, the transgenic expression of GCAMP3 is still somewhat lower than the AAV expression of GCAMP3, so it requires higher laser power to do this whole set experiment. But other than that, the response properties are very similar, comparable between the transgenic mice and AAV expressed GCAMP3. And of course, they have differential properties from the calcium dyes, OGB. Finally, just a few slides talking about our progress. Yeah, okay. I will actually stop here. So yeah, we are developing all kinds of intersectional strategies. I just wanted to show that in our most recent work, we have demonstrated that using a combination of parvalbumin flip or parvalbumin G with a VGAT Cre and do that in a double reporter, we are able to achieve pure parvalbumin positive interneuron expression. And this is, people know that the parvalbumin gene is also expressed at low levels in excitatory neurons. The labeling is never 100% pure interneurons, but by doing this kind of double intersectional strategy, we are able to actually label all the cells and only interneurons. And just one example showing there's no axon fibers seen crossing the white matter here. That's an indication that no excitatory neurons or at least layer 5 excitatory neurons are expressed. So I want to thank a large number of people that are involved in both types of projects, people at the Institute as well as a large number of collaborators that we have fortunately had, our advisory council on the Connectivity Atlas project, and all the different teams at Allen Institute, and of course our founder for their generous support. Thank you.